Thank you, Lynn, and thank you so much for inviting me. I actually see Lynn every day. We have a very nice plaque of Lynn in our clinic as the founder of the clinic, and I never got a chance to publicly acknowledge the enormous impact that you've had on dementia care in BC and beyond, as well as research in Canada and internationally. So I can't think of a more deserving person for the Order of Canada, Lynn, so congratulations. All right, now I get to try to use this thing. There we are. Okay, so a few disclosures. I am going to discuss a drug that's marketed by Biogen, although I don't think this is very relevant. So this was a clip from the New York Times about 6 months ago saying that it's a new exciting drug that may have an impact in Alzheimer's disease. This is very exciting, but we're going to talk about today is where did that drug come from and what process did it go through to become a drug? And it's a very interesting, complicated story, and I'll give you just a very brief snapshot of what that looks like. And everything starts with an observation. And the observation that's still relevant today actually happened more than 100 years ago, where it was discovered that patients who have dementia, and especially memory loss, sometimes have these things in the brain. So one is amyloid plaque. And I think if you're not familiar with this term already, I would strongly encourage you to become familiar with it because you're going to read about it in the news many times in the years to come. So A beta refers to a plaque in the brain or something soluble in the blood, but A beta you should be familiar with, means amyloid. The other observation was neurofibrillary tangles, and there's many other things going on in the brain in Alzheimer's disease, but these were the key observations that eventually led to some drugs that are coming out now. The second observation was that in Alzheimer's disease, the brain seems to shrink, and it shrinks much faster than someone who doesn't have Alzheimer's disease. And we think that's because nerve cells and other supporting cells in the brain, they die over time. So that formed, those two observations really formed what is known as the amyloid hypothesis. And that is a gradual buildup of amyloid in the brain, or A beta, leads to nerve death and brain shrinkage, and that causes Alzheimer's disease. And when you have a hypothesis like that, you take it into the lab and you test it, and that's how drugs really eventually make it to patients. So the major hypothesis to be tested in the lab was, first of all, is amyloid or A beta toxic to neurons? And of course, neurons are a major component of brain, and we think that's why it shrinks, because you lose neurons. And the beauty of science is that you can try to distill the brain, which is enormously complicated. You can try to distill it into its most functional components. And of course, one of the most functional components is the nerve cells. And we've known for decades now, and we have beautiful methodology to actually grow these cells, these individual neurons, in a dish. And this is what it looks like. I love looking at these pictures because I think it's just so beautiful. These are, again, we have billions of these in the brain. And these are individual nerve cells that, again, constitute the majority of what the brain mass is. And we can culture these from animals, from mice. We can even culture them from humans. But we can do even better. So if you look at a very small component of the nerve, you see that little tiny red box there on the top? You can look at a little segment of neuron. And these are called dendritic spines. That's what those arrows are pointing to, those little clumpy things that are sticking out. And those are the individual contact points between neuron to neuron. And this is probably my favorite picture, actually, in all of neuroscience, because these connections determine who you are. They encode memories, they retrieve memories, they determine your personality, they make me talk as I'm giving this talk. Those are connections between these tiny dendritic spines. So the experiment in the lab with the Lecanemab, which is this new drug that's coming out, or any other drug, was whether A beta was toxic to these very basic elements. And what you can do with these dendritic spines, or these cultured neurons, is you can observe them over time. So you can imagine having something growing in a dish or something going on anywhere, and you have a camera that looks at it, and you can look at it constantly for 5 hours and see what happens. And the beauty of that is you can look at individual dendritic spines and you can see what happens to that little guy over the course of 5 hours. And what you can see here is, so the picture on the left and the picture on the right is the same segment, but can you see on the right that some of those little spines are gone? So that's after you apply amyloid or A beta onto that culture. So it becomes toxic and you lose dendritic spines, you lose contact points. And as this goes on over time, we think it impacts how you retrieve memories, it impacts how you encode memories, it impacts everything. So the model is, and this is not a perfect one-to-one relationship. I wish it was, but it isn't. But the idea is that the condition on the left kind of resembles a normal brain. And when you add A beta, you lose dendritic spines. Over time, you lose whole segments, and that leads to the brain shrinkage that you see on the bottom there. So in order to target A beta, you need to understand just a little bit on how it works. So at the bottom there, you can see something attached to a nerve cell. That's what that meant to be at the very bottom left. And this is something we all have. So A beta is something we all have in our bodies. It just happens to go a bit awry in Alzheimer's disease. And there's two types of A beta that's particularly toxic. One is that little assembly there that you see, A beta oligomers. It's a little confusing term, but those are fairly toxic and they act on cells through a receptor. That's what that other guy sticking out the cell is. That's a receptor for A beta. And the other thing it does, it clumps up into plaques, which I showed you already. There's so many plaques in the brain. There's 60 million deposits of plaque in someone who has Alzheimer's disease. So it's a big problem. So if you're a drug developer, you can pick a couple of these pathways. So you can say, well, I'm going to eliminate that and see what happens if that's an effective therapy. And we'll get to that a little bit later, because that's where some of the drugs that are coming out now are targeting. Or perhaps you're in a different mood and you want to target the receptor for A beta instead and see what happens. So I'm going to give you an example. This is some of the work we did before, what happens when you take away the receptor. But you can really go in any pathway here and test it. And that's what any drug that you see come to market or come to a patient goes through this process. So here's back to our neuronal segment again. The test now is, here's another segment where no A beta is applied. So now we're going to apply A beta again, and you're going to look at those individual dendritic spines there and see if they're any different. But the only difference now is we've removed this receptor for amyloid. So if it works, you shouldn't see an impact of A beta on the nerve cell, right? And you don't. So if you want to take a little time to study this, we spent hours studying this to make sure that there wasn't anything we're missing. But they all seem to be there, in contrast to the condition on top, where if you add A beta, you lose dendritic spines. If you remove the receptor for A beta, you don't seem to get an impact. So this tells us that we're onto something. So maybe we can find a drug that blocks that receptor, and we want to test it further to see if we can make it into a drug. And that's exactly what happened with some of the drugs you're seeing today. So the next step is you have to test this in a mouse model. And I'm always a little wary of showing this in a public forum, but the truth is animal research is an integral part still of dementia research and drug development. And I think we should be very transparent about that. And it turns out that if you increase A beta in a mouse, its memory becomes very poor. And that's what I'm going to show you what happens here. So they're in a little pool and they train to find the platforms because they want to get out of the water. They train to find a platform that you can't see, but you'll see it in a moment. And we compare a mouse that is sort of normal and one that is an Alzheimer mouse and someone where we've increased A beta. So this is the normal condition. You can see this guy here. There's the platform. You didn't see it, but the mouse found. So it's been trained to do this. Now here's, in contrast, a mouse that has quote unquote Alzheimer's disease. And this is in some way similar to someone with dementia having difficulty finding their way around in environments. You can see it takes much longer and eventually we'll find it. So if you just draw a line, you can see the difference. It takes much longer for a mouse to find this platform. And this is really the basis for which drugs are tested. Can you find a drug? In this case, we showed that removing the receptor for amyloid seems to work. So if we do that in a mouse, can we eliminate this pattern? So we can graph this very briefly, the blue bar there. So over time, this normal mouse takes less and less time to find the platform, in contrast to the red line where there doesn't seem to be any learning. So if this drug works, you want it to be more similar to the blue line and not so much to the red line. And that's what happens. So if you remove the receptor, you seem to cure Alzheimer's in a mouse. Now, we've cured Alzheimer's in a mouse hundreds of times, and that's one of the problems with this model, is that it's difficult to translate those findings into humans. But this remains a very important part of drug development for any drug that's going to make it to a human. But for this reason, because it's so difficult to translate, it turns out, maybe not a surprise, that mice are not people. We're trying some different approaches. And one of those different approaches is to take human cells and model those in a dish in a more complicated three-dimensional environment. And I'll show you some of that data. But the beauty of this is just a maybe 10-year-old methodology. So you can take blood now. You can go to a lab, any of you, and you can have a blood draw. And we can reprogram your blood cells into your brain cells. And we can make little mini brains, your little mini brain, in a dish. So these are from a person. These neurons are reprogrammed from blood cells. So they're in Alzheimer patients' own brain cells. And we can make different kinds, not just neurons, which you see in the middle there, but we can make other supporting cells called astrocytes. And there are other, again, subtypes called microglia. Those are the three types of cells we like to make and try to model. And you can make three-dimensional structures. So this is a 3D, so you've all heard of 3D printing, but this is a 3D bioprinter where you take those cells and you actually print little brains. So you construct little brains in a dish. So that's what you're seeing here, is a very precise deposition of cells. This is just a test construct, but we're trying to make a 3D environment that better mimic the brain environment. You can see it better here. These are just deposited in a multi-layered structure using 3D printing technology. And all the cells are in this red goo here that gets deposited. And this is what it looks like. So they are kind of balls of tissue. And the surprising part is when you add other cell types in with these structures, these are microglia. This is very new. No one's ever seen this before. If you add microglia or supporting cells into these 3D structures, these microglia go in and out of the cell structure to try to find something that's abnormal. In this case, it's A beta it's looking for inside the structure. So we hope that these 3D models will allow us to use less animal work as we develop drugs. And we also hope that this will allow us to probe mechanisms that we simply can't see in animal tissue, but we can see it in cells that come from patients themselves. And then finally, I'm going to just touch on Lecanemab. So most of the news articles you're going to read in the next year and a half or 2 years are antibodies against amyloid. So that's what this pathway is. And the beauty of this is really amazing. I would say of the most remarkable findings in the entire Alzheimer field in the last 10 years, I think it's this. It doesn't mean that we're necessarily very close to a cure, but we've come a long way. So this is a PET scan. So you can think of a PET scan as just as a CT scan, and it's specialized to look for amyloid in the brain. So in this case, when it's very red, you have a lot of amyloid. And when it's not so red, which you see on the right, you don't have a lot of amyloid. This is actually the same person here, but it's 1 year after being treated with one of these antibodies. So you can now take, be it Lecanemab. Lecanemab is this new drug that has come out. There's about 6 of them now. If you take any of those for a year, you can completely remove amyloid in the brain over the course of 12 to 18 months. That's what you see here. It's really remarkable. So now you can really test, if you remove amyloid in the brain, does that cure Alzheimer's? And that's what, of course, everyone is trying to do. And I'll leave you with this. This is the data from Lecanemab. We want to discuss that drug in detail, but I'm happy to take any questions after this. So on the left side, you just see how it has a profound impact on amyloid in the brain. So on top, you see Placebo, which means someone who has received a similar injection, but it doesn't contain the actual drug. So it's like a sugar pill. And you can see that their amyloid in the brain doesn't change over time. This is 18 months. But if you take in the orange there, if you take Lecanemab, you see a very dramatic reduction where brain amyloid is normalized over the course of 18 months. And on the right, that's the real critical piece. So they looked at a cognitive measure. And it turns out that at 18 months, if you look at the orange line, so in this case, lower is better. They have a slightly less decline. They estimate it to be about 27% over 18 months. Now, mind you, and this is the, I guess, reality check, you'll see in the news that it's 27% reduction in Alzheimer progression, but the change is very small. In fact, the change is so small, I'm not convinced we can measure this in the clinic, but in a study with thousands of patients, you can detect very small changes. So it's progress, but we're not quite there yet. And with that, I will thank you for your attention and just some collaborators.